Hello, my name is Gisela Umbuzeiro, and I'm going to present the work Parhyale hawaiensis as a key species for ecotoxicity evaluation in marine tropical environments. Well, we felt the necessity of model organisms from marine ecosystems to work with ecotoxicology. We found this organism as a potential species, Parhyale hawaiensis. Parhyale is already a model in genetic and embryonic studies. It's very easy to handle in the lab. It's an epibenthic organism and has a worldwide tropical distribution in the marine area, especially in shallow waters. So we thought maybe this could really be a suitable model for ecotoxicology. In fact, we have been working with this organism for around 10 years now, and we developed a portfolio of protocols. And I'm going to present them in detail further on. First, regarding culture testing, we defined culture conditions, acute toxicity tests for water and sediment samples, chronic toxicity tests based on reproduction and growth, and antenna regeneration tests. We also thought it would be very neat to have protocols for internal dose. So we learned how to extract hemolymph from adult animals. Also, we developed feeding exposure conditions, very important to some types of materials to be tested, like nanoparticles, especially thinking that it's an epibenthic organism, so it would be interesting to see how feeding exposure would work. And we also developed a method for analysis of silver and copper in the hemolymph. Regarding genotoxicity and gene expression, comet assay and micronuclei where the protocols were defined. Also, we defined the protocol for RT-qPCR for glutathione S transferase and global gene expression using RNA-seq. Immunotoxicity, hemolymph cells characterizations are still under development, and immuno response to yeast verification also is being performed right now. Culture and acute toxicity test. Basically, the papers were published by Artal, as you can see on the bottom of the slide. Artal 2018 and 19, and also Vacchi 2019. So successive generations have been successfully cultivated in the lab. Protocols for acute toxicity test with a miniaturized protocol in 96 wells. It worked very nice with a single neonate in each well. Acute toxicity test responses were in the same range of sensitivity of other amphipods. A miniaturized protocol for sediment that was successfully applied and confirmed toxicity from samples under the influence of a sewage outfall in Santos Bay. Also, we developed a miniaturized protocol in 12 well plates. Internal dose, this work was done by Vannuci-Silva et al. We developed two types of protocols using atomic absorption and ICP-MS. Both provided satisfactory results in the quantification of silver and copper in the hemolymph. As you can see on the left, hemolymph is being taken off the adult animal, and you see it's kind of a beautiful blue. Silver concentrations in the hemolymph increased in relation to the increase in concentration of silver concentration in the water. We did a very nice toxicokinetic study. Silver bioavailability was higher in organisms exposed to silver nanoparticles via feeding, indicating that the nanoparticles pose a higher risk of toxicity compared to similar concentrations of the diet containing silver salts. Gene expression, This was a lot of work, but also Artal and colleagues. The expression of transcripts of GST did not differ between AG South and AG nanoparticles treatments or over time. Then we decided to go, it was a little bit disappointing, then we decided to go for the whole genome transcriptional differences, and we found very important difference between males and females when we observe, we exposed the animals also via feeding to the salt and silver nanoparticles. The silver nanoparticle treatment resulted in an increased number of genes differentially expressed in both sexes. Males presented twice the number of differentially expressed genes in comparison to females, although from the same biological process. The main finding is that when we are going to do gene expression, apply it to toxicology in those organisms, it's important to take into consideration the differences between sexes. Genotoxicity, this work is still ongoing. This was done by Botelho, Marina. Protocols were optimized because it's a marine organism, so we tested different types of buffers and cell preparations. Finally, we got a good protocol. Then we did some experiments, exposed organisms to silver nanoparticles through feeding again, and we found a significantly higher micronuclear frequency in the HEMOLYMPH after 9 days of exposure. When we did DNA damage, we also optimized the protocols for COMET assay, but we didn't see any difference. Chronic toxicity test, it's also ongoing now. This is mainly done by Amanda dos Santos and colleagues. The best conditions were reproduction and growth. This is a lot of work. It was very challenging to start this chronic toxicity test development. Finally, we think we have a good protocol now, especially the time frame that we are going to monitor. So we now are going to work with organisms that are 52 days old. They will be followed for 40 days, neonates recorded 3 times a week, and growth rate will be evaluated in time 0 and time 40. We believe this is going to work, and now we are using zinc and copper as reference substance to see like a proof of concept of our protocol. Another test we developed, this was very interesting. It was the antenna regeneration test done by Otávio Diel and colleagues. We had a very important contribution of Henrique Marques from University of Campinas and Tiago as well. So the test conditions for antenna regeneration was developed. The total length of the test was 35 days. The test consisted in cutting one of the antennas of the Parhyale, and then you can see in the picture with the bee that this was cut, and then we waited for the complete regeneration of the antenna. This was very, very elegant, I think, and we were able to test as a proof of concept. We tested the growth inhibitor pesticide diflubenzuron, and this compound inhibited antenna regeneration with an EC50 of 0.5 micrograms per liter, so it's in the PPT level. This was very interesting. We are now preparing the paper for submission. Finally, immunotoxicity. Well, we wanted so badly to go for this endpoint because all the importance of working with particles as nanoparticles and also as microplastics. But then we needed to characterize the HEMOLYMPH cells first, right? Because nothing has been described in the literature yet. So conditions for characterization of living HEMOLYMPH cells were established. Good results were obtained with slides preparing with cytocentrifugation and Rosenfeld coloration. Sorry for the pictures, they are not very good because we just did those experiments, and now because of the pandemic, we could not make better pictures. But you can see on the right, you can see a cell that has more granules, and also on the left, cells without granules. So we believe we have for sure two types of cells, and maybe two types of granular cells we are going to see. Unfortunately, flow cytometry did not work because the amount of hemolymph is very small. We only obtain per animal like 0.5 microliters of hemolymph, so we couldn't do it. As conclusions, we are very happy with this Parhyale hawaiensis. If the question was, is Parhyale hawaiensis a good model in ecotoxicology? Our answer is yes. It is a suitable and reliable organism for ecotoxicological studies. It's very easy to grow them in the lab. They really like the lab. It's amazing. And they are so cute. A portfolio of protocols are available now and can be used to expand the use of Parhyale hawaiensis as a model test organism representing marine tropical environments. So thank you very, very much for your attention. And I want to acknowledge the universities that gave us a lot of support, CAP, CNPq, and FAPESP for funding. Thank you very much.